They say breakfast is the most important meal of the day, that the body requires constant fuel, 3 meals, snacks between them, a steady stream of nutrition to maintain energy, metabolism, and focus. That eating less frequently risks muscle loss, metabolic slowdown, and nutritional deficiency. These beliefs have shaped public health guidance for decades. And yet, for much of human evolutionary history, eating once or twice a day was not an anomaly. It was the biological baseline. The body was not designed around a 12-hour daily feeding window. It was designed around feast and fast, around extended periods without food, followed by concentrated nutrition. OMAD, 1 meal a day, compresses all caloric intake into a single meal and a feeding window of approximately 1 hour, leaving 23 hours in a fasted state. What that compression does to the body's internal biology, its hormonal rhythms, its metabolic machinery, its cellular housekeeping systems, is measurable, documented, and considerably more complex than either its proponents or critics typically describe. This is the science. Let's begin. Insulin, the hormone that controls everything. To understand what OMAD does to the body, it is necessary to first understand what happens every time food is eaten. Every macronutrient, carbohydrate, protein, to a lesser extent fat, stimulates the pancreatic beta cells to secrete insulin. Insulin's primary job is to facilitate the movement of glucose from the bloodstream into cells, where it can be used for energy or stored as glycogen in the liver and muscle. Insulin also suppresses lipolysis, the breakdown of body fat, and promotes triglyceride synthesis and storage in adipose tissue. In a standard eating pattern involving 3 meals and 2 snacks across a 12 to 16-hour daily feeding window, insulin is elevated for the majority of the waking day. The body spends most of its time in a fed, storage-oriented metabolic state. In this state, circulating fatty acids are not being mobilized for fuel. Insulin prevents it. The body's access to its fat stores is gated off by persistent insulin signaling. Cellular systems that are activated in the absence of insulin, autophagy, fat oxidation, certain repair pathways, are chronically suppressed. OMAD reduces the daily insulin profile to a single peak. One spike following the meal, followed by 23 hours of progressive insulin decline. During those hours, as insulin falls toward its baseline, the body's metabolic state shifts. Glycogen stores in the liver are gradually depleted over the first 8 to 12 hours. As stores decline, the liver shifts toward fat oxidation. Circulating free fatty acids rise. The metabolic machinery responsible for burning fat, rather than storing it, becomes progressively active during the extended fasting period. The cellular environment changes fundamentally when insulin is absent. What happens in that environment is the biological story of OMAD. Ketosis, the liver's secondary fuel system. Approximately 12 to 16 hours after the last meal, a metabolic threshold is crossed. Hepatic glycogen, the glucose stored in the liver, is approaching depletion. Blood glucose levels fall toward their lower physiological range. Insulin remains low. The body requires fuel, and it activates its secondary fuel production system. In the liver, the enzyme HMG-CoA synthase initiates the conversion of acetyl-CoA, derived from the oxidation of fatty acids, into ketone bodies: acetoacetate, beta-hydroxybutyrate, and acetone. These molecules are water-soluble and can cross the blood-brain barrier, a property that glucose shares, but that most other fuel molecules do not. The brain, which in a glucose-dependent metabolic state consumes approximately 20% of total body energy, can derive a significant proportion of its ATP from ketones during extended fasting. Neurons adapt to ketone utilization over days of consistent fasting, are increasing the expression of the transporters that carry ketones across the cell membrane. Beyond their role as an energy substrate, ketones function as signaling molecules. They inhibit NLRP3, an inflammasome protein involved in pro-inflammatory signaling. They activate HDAC inhibition pathways that influence gene expression, and they stimulate the production of brain-derived neurotrophic factor, BDNF, a protein that supports neuronal growth, synaptic plasticity, and the maintenance of cognitive function. The liver during the OMAD fasting window is not idle. It is running an entirely different fuel economy. Autophagy, the cell's internal cleaning system. Beyond fuel production, the extended fasted state of OMAD activates one of the most consequential cellular processes in human biology, autophagy. The word comes from the Greek for self-eating, and it describes precisely what occurs. Cells identify damaged or surplus internal components, proteins that have been cross-linked or misfolded, organelles that are no longer functioning at capacity, pathogens that have been internalized, and envelop them in a double membrane structure called an autophagosome. The autophagosome fuses with a lysosome, which contains digestive enzymes. The contents are broken down and the molecular building blocks are recycled into new, functional cellular components. The primary trigger for significant autophagy activation is a drop in intracellular nutrients, specifically the fall in amino acid and glucose levels that occurs during extended fasting. Approximately 16 hours into a fast, when autophagic activity is substantially elevated compared to the fed state, AMPK, the cellular energy sensor activated by low ATP, promotes autophagy while simultaneously suppressing mTOR, the kinase complex that drives cell growth and protein synthesis, and that inhibits autophagy when nutrients are abundant. For mitochondria specifically, a form of selective autophagy called metaphagy removes dysfunctional mitochondria that are producing excessive ROS or no longer generating adequate ATP. This quality control process is one of the primary mechanisms by which the mitochondrial population within a cell is maintained in a functional state over time. The OMAD fasting window, lasting approximately 23 hours, provides consistent daily activation of this cellular housekeeping system. Metabolic flexibility. What the body learns. A healthy metabolic system is not one that runs exclusively on a single fuel. It is one that can access and efficiently utilize whatever substrate is available. Glucose when carbohydrates are present, fat and ketones when they are not. This capacity is called metabolic flexibility, and it is measurably diminished in individuals who spend the majority of their time in a fed, insulin-elevated state. When the body is consistently exposed to dietary carbohydrates across a long daily window, the enzymatic machinery responsible for fat oxidation, the proteins involved in fatty acid transport, mitochondrial beta oxidation, and ketogenesis, is chronically underexpressed. The cells are, in a functional sense, specialized for glucose combustion and poorly prepared to switch when glucose is unavailable. This metabolic inflexibility is associated with, among other outcomes, significant mid-afternoon energy crashes when blood glucose falls between meals, and the reflex dependence on frequent eating to maintain energy stability. OMAD, by definition, alternates daily between a concentrated fed period and an extended fast. The consistent daily transition through the metabolic shift, from glucose to glycogen depletion to fat oxidation to ketogenesis, trains the cellular machinery responsible for each of these states. Enzyme expression for fatty acid transport and oxidation increases. The mitochondrial response to falling glucose improves. The body becomes more efficient at recognizing and utilizing the metabolic state it is currently in. The myths around OMAD and muscle loss. The most persistent concern raised about OMAD is the loss of muscle mass. The conventional model of sports nutrition holds that muscle protein synthesis requires frequent protein feeding, that without amino acid availability throughout the day, the body enters a catabolic state and breaks down muscle for fuel. The biology is more nuanced than this model suggests. The rate of muscle protein synthesis is not determined solely by the frequency of protein intake. It is determined by the total leucine threshold, the concentration of the amino acid leucine in the bloodstream and muscle cell, which acts as the primary stimulus for mTOR activation and subsequent protein synthesis. A protein-dense single meal delivers a substantial leucine stimulus, one that can activate muscle protein synthesis to a degree comparable to multiple smaller meals, provided the total protein content is adequate. Research examining body composition in individuals practicing alternate day fasting and time-restricted eating has consistently shown that lean mass is largely preserved when protein intake is adequate. even when total caloric intake is reduced. The direction of the body's substrate preference during fasting is toward fat, not muscle. Muscle loss under OMAD is not an inevitable physiological outcome. It is a risk associated with inadequate protein intake within the meal, a nutritional variable, not a biological certainty. So at the risks. What biology also shows. The biological changes associated with OMAD are not uniformly beneficial. The same physiological parameters that create its metabolic advantages also create specific risks when the dietary context is not appropriate. The most immediate risk is digestive overload. The gastrointestinal system is designed to process food that arrives in portions across a feeding period. When the entire day's caloric intake arrives as a single large bolus, the mechanical and enzymatic capacity of the stomach, small intestine, and digestive enzyme production may be acutely overwhelmed. Gastric stretching beyond normal capacity activates pressure receptors that produce bloating and discomfort. Even accelerated gastric emptying in response to an unusually large meal volume can produce reactive hypoglycemia in the postprandial period. Acid reflux risk increases with meal volume. Nutrient adequacy is a significant concern. Meeting the recommended daily allowances for micronutrients, vitamins, minerals, trace elements, in a single meal requires careful dietary composition. In practice, individuals who adopt OMAD without nutritional planning are at risk of chronic deficiencies in vitamins D, B12, calcium, iron, zinc, and magnesium, each of which has documented physiological consequences in the tissues and systems they serve. Specific populations are contraindicated for OMAD on physiological grounds. Pregnant women require consistent nutrient availability throughout the day for fetal development. Children and adolescents in active growth phases have continuous anabolic demands that a single daily meal cannot reliably meet. Elderly individuals are at heightened risk of sarcopenia, age-related muscle loss, and require consistent protein distribution to preserve lean mass. OMAD is a powerful metabolic tool. Like all powerful tools, its effects depend entirely on the precision with which it is applied. What the body is telling you. Signals from the fasted state. For most individuals beginning OMAD, the first 1 to 2 weeks involve a consistent and recognizable set of physical experiences. Understanding what these experiences represent biologically transforms them from obstacles into information. The hunger experienced in the initial days is, in large part, conditioned hunger, not physiological starvation. Ghrelin, which the stomach-derived peptide known as the hunger hormone, is released in anticipation of habitual mealtimes. The body has been trained to expect food at specific hours. When that food does not arrive, ghrelin is secreted on schedule. The hunger is real in the sense that the hormone is genuinely elevated. But over 10 to 14 days of consistent altered timing, ghrelin secretion reprograms to align with the new schedule. The habitual hunger at former mealtimes diminishes. The mental clarity that many individuals report during the fasted state, typically emerging after 2 to 3 weeks of consistent practice, corresponds biologically to ketone availability in the brain, elevated BDNF, and the reduced neuroinflammatory signaling that accompanies lower insulin and reduced dietary ROS production. On each of these signals is the body communicating its current biological state. They are not discomforts to be suppressed. They are the physiology made visible. OMAD is not a diet trend or a willpower test. It is a specific pattern of caloric timing that produces measurable, documented changes in the body's hormonal environment, metabolic machinery, and cellular maintenance systems. What the body experiences during 23 hours without food is not deprivation. It is a biological state, one that activates systems for repair, fuel flexibility, and cellular housekeeping that are suppressed by continuous feeding. Whether those changes are beneficial, neutral, or potentially harmful depends entirely on the individual's physiology, the nutritional quality of the single meal, and the broader context of health. When this channel explores the science behind everyday health topics, and if you want more simple tips to improve your health and mind, make sure to subscribe, and I'll see you in the next video.